Bismillahirrahmanirrahim. My dear students and dear viewers, Assalamu alaikum warahmatullahi wabarakatuh. Welcome back to AmreenaMehrajTutorials, and I'm very excited and happy that I'm in front of you today again with this very important topic called hemophilia. My dear students, hemophilia is a genetic disorder. I've already given you the classification in my previous lectures. I've already made you understand what basically genetic disorders are and how you can classify them. Now we have to focus in upcoming lectures. I have to be focusing upon important genetic disorders. Number one genetic disorders I've selected for today is hemophilia, as per the suggestions I received. So hemophilia falls under which category? It's a sex-linked recessive disorder. This is the first thing you have to remember. That means this disorder is related to some kind of problem in the sex chromosome. And specifically which sex chromosome it's going to be? It's the X chromosome. So we should say it's an X-linked recessive disorder. Okay? Or you can remember sex-linked disorder, because later on in inheritance pattern or at the time of mutation, you'll click that the mutation has taken place in the X chromosome. That's why we use the word X-linked or X chromosome-linked recessive disorder. Now, my dear students, before we start this lecture, I just want you people to give this video a like, a heart, a share and a subscribe if you find it worthy. Okay? And keep on supporting. Your support is really a lot. And now, my dear students, let us go back to the disease, the disorder to be specific. Hemophilia is a very interesting disorder, and you are asked questions out of this particular stuff. Before starting, I just want you people to look at the textbook. I do have this NCERT textbook with me. I don't know whether you will be able to have a look on this or not, but still, since textbook is very important, I want you people to realize its importance. So we are going to go into the depth as per the CBSE standards, but don't forget to check out your textbook. That's very, very important. So we are currently going through Mendelian disorders, the basics, the pedigree symbols. I made you understand that in my previous lectures. Now we will go to the diseases one by one. We will not discuss only these diseases which are in the textbook. We'll go beyond this. We'll go beyond the explanation which is on the textbook, but you have to have a hold on your textbook. That's very, very important. Okay, let's get back to the work. So hemophilia, my dear students, it is otherwise also called bleeder's disease, and it's also called, let me go down the alternative names first of all, it's also called royal disease, because there is a lot hidden in these alternative names. Okay, look, from the bleeder's disease you got this idea that the disorder is pertaining to defective blood clotting. You got this idea just from the alternative name, because the name is bleeder's disease. That means there will be some sort of defect as far as blood clotting is concerned. What is blood clotting? Basically a mechanism in which if we have an injury, then we have a clot to be formed. That clot formation takes place by clotting mechanism. Now, if there will be some defect in the clotting mechanism, that means we will continuously bleed. So that is the case. The defect in this particular disorder lies in the clotting mechanism. Let me make you understand now this thing in detail. Let us go in explaining what basically this disorder is. So this disorder, my dear students, in this disorder, a person continuously, this is the word I must use, a person continuously bleeds. That means bleeding keeps on going on. Even from a minor cut, I can see. That is the dangerousness. Just a minor cut, just a slight cut. We experience slight cuts on the body, abrasions and bruises. But they are very dangerous to a person who is suffering from hemophilia. Because if he or she will have a slight cut on the body, they will keep on bleeding. Till death, they can bleed. That could be the condition, although, okay. Now, my dear students, what can you guess from over here? What could be wrong with these organisms is, I already told you, they cannot form a clot. Let me make you understand. What happens in normal cases? Normally, what is the situation? Although our topic is not blood clotting, but let me make you understand. Normally, suppose this is your blood vessel. Okay, this is one of your blood vessels in the body. Suppose you experience some injury. Okay, once you get injured, there is a kind of injury to the blood vessel. Now, if blood vessel gets injured, what is flowing inside is your RBCs, WBCs, platelets, plasma, your blood. Now, the platelets which are over there, automatically, I will not be going to the mechanism part. I'll make you understand over here that the platelets immediately will aggregate near this injured part. This aggregation of the platelets will result in the formation of a platelet plug. This is the first thing which will happen. Other than that, this blood vessel will itself constrict, so that lesser amount of the blood will reach to this particular area. It will constrict. So constriction or constrict, I must say constriction, I must write down constriction of blood vessel. These are the normal things which happen just to prevent the loss of the blood. Okay, this is the under normal physiological conditions this will happen. And then once you have this blood platelet plug formed, then inside what will happen is a blood clotting cascade of reactions. That means there will be now certain chemicals which will be activating other chemical, which will be activating other chemical. Finally, you have to basically convert fibrinogen into fibrin. Okay, these fibrin threads will then get aggregated over here. So what are these lines which I am making? This is the final fibrin clot, which is very, very strong. This clot is going to be it's going to be a strong clot which will basically prevent the further loss of the blood. Now, what am I talking over here is this particular cascade of reactions. Over here, what will participate now are blood clotting factors. They will participate, one will activate, another will activate another, and in that cascade, finally, their purpose is to convert fibrinogen into fibrin. This is what happens normally in blood. And how many blood clotting factors are particularly over there if we take into consideration both of the pathways? Because we have pathways of blood clotting. We have the intrinsic pathway and we have the extrinsic pathway. Taking both into consideration, we have 13 blood. These 13 blood clotting factors are required for the formation of what? What is the target? The target is the fibrin clot. Okay. Then once fibrin is formed, then fibrin clot, a strong clot is formed, that will assure that the loss will not take place. Blood loss will not take place. Now, what is the problem in case of hemophilia? What is the problem in case of hemophilia? What is the problem? That there is problem with the blood clotting factors. You have basically what? You have a kind of deficiency of blood clotting factors. Now the question arises, ma'am, is it like all the 13 blood clotting factors are not present in this individual? No. There are certain blood clotting factors which are not present. Accordingly, you have the types of hemophilia. How you can think? If a particular clotting factor is not present, if a particular clotting factor is absent, what will happen? Will this fibrin clot be formed? No. If a fibrin clot is not formed, can this fibrin plug will be formed? No. When we have no fibrin fibers, fibrin clot will not be formed. When fibrin clot is not formed, is a strong kind of plug formed? No. What will happen to the blood? Blood loss will keep on going. It will keep on going. That's the condition we say that the person will continuously bleed. Now let me make you understand this in further detail. So you got to know this idea first of all, why it is called bleeder's disease, because there is a problem in the blood clotting mechanism. Okay, if you wanted to go back to the this description, you can go back and take a pause and you can copy that. Okay, now my dear students, you got this idea that what is the reason is the defective blood clotting mechanism. Clear to you people? Accordingly, can you agree now? It could be called bleeder's disease. Okay, now my dear students, what else we need to remember is this problem, this defective blood clotting mechanism is caused due to what? Can you guess? It is caused due to deficiency of blood clotting factors. I'm sorry my handwriting is very weak, but still I'm speaking and that will help you. Okay, so there's a deficiency of blood clotting factors. Now, according to the type of which blood clotting factor is deficient, we have the types. So you have to remember totally how many clotting factors. I'll use CF apostrophe for clotting factors. We have 13. Now, if there is the deficiency of clotting factor, I'll write down factor. We have the numbers now. If there is a deficiency for factor 8, we use the Roman numerals. Deficiency of factor number 8. This factor is also called anti-hemophilic globulin. Okay, this is the name of this factor. If this factor is deficient, then the hemophilia is called Hemophilia A. It's called Hemophilia A. Another, so we have either the deficiency of this clotting factor, another if we have the deficiency of factor, clotting factor number 9. I hope I'm using the correct Roman numeral. If there is the deficiency of blood clotting factor number 9, then let me give you the alternative name of this blood clotting factor. This is plasma, this is very important, thromboplastin cofactor. Okay, or it's also called, let me give you the alternative name, it's also called Christmas factor. If this factor is present, it was basically discovered in a Christian family, that's why. Then this particular hemophilia, because again you will have a defect in clotting mechanism, again it will be a bleeding condition, again you will call it a hemophilia. This hemophilia is called Hemophilia B. Okay, let's for the time being consider these two cases. I'll give you later on another type. Okay, so you have Hemophilia A, Hemophilia B. Okay, Hemophilia B is also called Christmas disease. Now you can think why it's called Christmas disease, because the factor which is absent is called the Christmas factor. And again, another thing is that it was discovered in a Christian family. That's why you call it Hemophilia B. Now, my dear students, you got to understand why bleeders disease. Let me give you an idea why royal disease. Royal word means the family, a royal family. Since this disease, this particular disease, it was investigated and recognized that it is a hereditary disease. It can run in families. That was first of all recognized by a physician. So let us remember the name of the physician, the doctor, which basically recognized what? He recognized that what? Hemophilia is a hereditary disease which runs in families. He also said that it affects mainly what? It affects mainly males. We'll also understand why it affects only males. That's the secondary question. This physician who first of all recognized this hemophilia is a hereditary disease which affects only male. Who was this physician we are talking about? This physician is very important to remember. He is John Otto in the year 1803, if I am correct. Yes, 1803 was the year when he first of all recognized that hemophilia runs in families. It's a hereditary disorder, and he basically studied its pedigree. He studied its inheritance pattern and said that this disorder will definitely affect the males in the family. Why only males? Why not females? I'll make you understand that also. This is going to be our next part. For the time being, what you understood is basically hemophilia is a bleeder's disease. It's also called royal disease. Bleeder's disease because in this disease, person will continuously keep on bleeding, even just through a minor cut, which is otherwise very normal. Okay, because we, alhamdulillah, Almighty Allah has kept this blood clotting mechanism in the body. Clotting factors get activated and we have a strong fibrin clot. But they do not form a strong fibrin clot because of defective clotting mechanism. Why defective blood clotting mechanism? Because there is a deficiency of what? The blood clotting factors. Now, which are the blood clotting factors which are deficient? In this case, we have two factors which are deficient. Either we do not have, I mean to say these individuals, because I'm not suffering from this disease. Either hemophilic individuals have clotting factor 8 absent, either clotting factor 9 absent. Accordingly, you have Hemophilia A. Accordingly, you have Hemophilia B or Christmas disease. Now, why is it called royal disease? That's very important, because this disease, actually it was seen in the family of Queen Victoria. Now, who is she? She is the Queen of England. And it was seen that she was the carrier for the disease. And because of her, the descendants of the family also showed the disease. So, this was carried by the descendants. So, this particular disease runs in the royal family, the family of Queen Victoria. She was a carrier. She basically transferred this disorder to her daughters, and once they got married in some other family, the children which were born out of those daughters, the males basically expressed the disease and they were affected. Now, this inheritance we have to discuss in a moment right now. So you need to remember that why is it called royal disease? Because it runs in the family of Queen Victoria, which is a royal family of England. Victoria herself was not affected by hemophilia. She was just a carrier. That means mutation was there in her X chromosome. She then passed on this, this defect, this mutation, this disorder. She passed on this to her descendants. That means definitely this disorder you will find in royal family, Queen Victoria's family. It was credited as a royal disease. Now, my dear students, let me make you understand what basically happens in this disease as far as the genetics is concerned. Let me make you understand. Before that, I need to rub on the This is the interesting part now. Pay attention. Now let us understand what happens in this disease. Let us understand what happens in this disease at the genetic level, okay? And let us understand then later on why it is only going to affect the males. I'll make you understand that also. So what happens in this disease? Let us understand. So the first thing you have to understand is that this disease is going to happen because of a mutation in the X chromosome. That's very, very important. So I will quote down this sentence that this disease or disorder, I'll be specific, this disorder is caused due to, remember, mutation on, see, very important, which chromosome is involved? I have told you in the beginning also. Mutation on X chromosome. That means the gene, you have to think, you have to now critically think. The gene for what? Blood clotting factor. That gene is basically located where? That gene is located on, X chromosome. Now, if that gene undergoes what? Mutation, so single gene is involved. Consequently, what will happen? That the clotting factor will not be produced. That means the gene is mutated. That's why the clotting factor is not getting produced. That's why the person keeps on bleeding. You're getting my point or not? So, what is the reason at the genetic level is the mutation of the gene for blood clotting factors, which is located on X chromosome. This is very important. Let me say that gene, let us denote that gene as H, okay? So gene H is responsible for, coding what? Blood clotting factors. So if gene H is normal, things will be normal. But if it is what? If it is mutated, then the person will develop hemophilia. You're getting my point or not? So if I'm writing X chromosome like this, it's what? It's normal. Okay, but if I show X chromosome and I will place a small h over here, you have to guess that this is a condition which is showing you what? This is a condition where you have been shown that there is mutation in a gene which is coding for blood clotting factors. Now, why I use H? This is an indication of the development of hemophilia. Obviously, this individual will develop hemophilia. So, I'm writing small h means this is the condition where I'm showing that the gene is basically mutated. And you have to remember that it's a recessive type of mutation. You will understand that also. First and foremost, you must understand that this is the normal X. But when I show like this, I mean to say that the mutation on the X chromosome has happened. And which is the gene which is mutated is gene H, which is mutated. Gene H, which is coding for the blood clotting factor. That's very important. Where is my duster? So that is very, very important to understand. This is the normal representation. This, as far as my lecture is concerned, I will show this particular form where I'll be showing this. Okay, I'll be showing X raised to power H, which means that the hemophilic condition is over there, mutation has happened. Understand? Different books will show it in different ways. Okay. Now, my dear students, let us pay attention. Let us now understand one more important thing. Very, very important thing. Female has how many X chromosomes? 2. Okay. If there is mutation on this chromosome, that means female will be safe because she has the other chromosome to check the expression of this defective X chromosome. So female can be saved out of this situation in this case. Okay, I'll give you the genotype separately. Okay, but look at the male. Male has only one X chromosome. Since male has only one X chromosome and this chromosome bears the gene for blood clotting factors, if male develops the hemophilic condition, there is no chance for male to be safe. He will definitely be a hemophilic male. Getting my point or not? So in female, there is a situation. If she has mutation on one X, she is safe because the other homologous chromosome will save her. The other homologous chromosome will form the blood clotting factors because that gene is correct. But male has only one X chromosome. What is the problem? Male has only one X chromosome. So if there is a mutation for gene H in male, male is not going to be safe because the homologous chromosome is absent. He has Y. Y doesn't bear this gene. And this mutation is going to get expressed in the male. Female is safe because the gene itself is recessive. Means the mutation form is recessive. The dominant gene will check the expression and she will be able to synthesize the blood clotting factor. So you got to know that since X chromosome is involved, so male will develop hemophilia. So who are going to be affected with these diseases? Generally, male is affected. Okay, female is safe in this condition. Female could also be affected. I'll make you understand this now, but for the time being, what is the conclusion you made is male will develop hemophilia because he has only one X chromosome. If mutation has happened to this X chromosome, male has no possibility. Male cannot produce then blood clotting factors because of the mutation. Getting my point or not? Now let us go and understand about the genotypes. Okay, so you got the answer what happens in this disease first of all. Okay, let us go to the genotypes. Now let us understand. This is now going to be very, very interesting. You will not be able to, you will not rather suffer in solving numericals or whatever will be asked out of this if you pay attention. So pay attention. Now, my dear students, think. If there will be defect in the gene, consequently, blood clotting factors will not be produced and the person will develop the disease. That means, where is the cause of the disease? The cause is in the mutation of the gene for blood clotting factors. Okay. Now, my dear students, let us pay attention to the possible genotypes. Here you need to have a strong hold on what homozygous, heterozygous, dominant, recessive means. Now, my dear students, as now you got this point that hemophilia is what now? You got this understanding. It's an X-linked disorder. And you want to understand that it will affect whom? Males. It will affect males. I will not say only males because I will make you understand now. It will affect males because they have only one X chromosome. Okay. Now look at these. Look at females. Females. In them, what is the situation? In them, what is the situation? They have two X chromosomes. Okay, because they have a homologous pair. If one X chromosome bears the mutation, this is which condition? This is which condition? This is a heterozygous condition. This is a heterozygous condition because here you have one gene mutated, one gene normal. In this situation, female will only act as a carrier because she has the mutation, but she will not develop hemophilia. Why she will not develop hemophilia? Your answer must be that, ma'am, she has this normal X chromosome. She has the normal dominant form of the gene. The normal dominant form will keep on what? That will keep on producing blood clotting factors. There will be no hemophilia. There will be no hemophilia. In which condition? Heterozygous. But ma'am, she is a carrier. Means if this defective X will be transferred to the offspring, get my point? If this will be transferred to the offspring via the ova, definitely the children has the possibility to develop the disease. Now, either male, either female. Think in critical manner. I will not say only male. You have to now think about it. Don't make it a rote learning. Use your brain. The other condition is XH. What is this condition? Female. But now the mutation has happened to the both X chromosomes. Condition has become homozygous. Condition has become homozygous as far as mutation is concerned. Both of the genes mutated. Now, in this condition, female will also develop the hemophilia because now there is no possibility for getting the blood clotting factors. But there is a point you have to notice that when could this female be formed? When could this female get birth? When both the father and the mother are defective. Means at least the father must be a hemophilic person, the mother must be hemophilia. That means only then these children will be born. This is very rare because generally hemophilic male do not survive for so long. And hemophilic, carrier mothers can transfer, but hemophilic male do not survive for so long a period of time. So chances of development of such an individual is rare, although this can happen. And number second thing is that this individual, this condition at the genetic level is a lethal gene condition. It's a lethal condition. Generally, these fetuses do not develop. This, development doesn't take place. It is discarded at the embryonic level itself. So these females you will not find out in population. Hemophilic females you will not find in population. So what is the point you got to conclude from this? If I put it in simple way, what is the point you got to conclude is that, ma'am, males, if they have XHY condition, if males have this condition, they will be hemophilic. Done. If females have this condition, they will not be hemophilic, they will be the carriers. If females have this condition, they will be hemophilic, but generally they do not take birth. Getting my point or not? Now we will study its inheritance pattern. So I have to rub off this board. Let me give you a quick, idea once again. So this is very easy, but as far as being capable of solving the numericals, you need to have in-depth knowledge. Only then you can solve the pedigrees and all, okay? So let us put it in a nutshell. X XX, she is what? A female. XX is a female, and if I write down H, she is a carrier. Okay, now if I write down like this, XX, both mutated, this is lethal combination. You will not see such females getting birth. Okay, and you say X and Y, this is a male who will be hemophilic. No possibility to be safe because only one X, that too mutated. No blood clotting factor will be produced in this case. Hemophilic male. So she'll be a carrier female. This will be a lethal combination of a female. Generally not born. And this is correct hemophilic male. So you want to conclude that who are basically going to be affected. Generally, if you look at the epidemiological level, at the survey level, who will you find in the population affected? Only males. Okay. Now let us look upon the inheritance. Let us take up the inheritance part of the disease. That is very, very important. That is how you come to know that why only males are affected. So I will give you the inheritance. Let me code down that particular inheritance which we'll study. This is which I'm going to basically take up now. That's called Crisscross Inheritance pattern. Very, very important. Crisscross inheritance pattern, or you call it zigzag inheritance pattern. You also call it a diagenic inheritance pattern, or you call it a diagenic inheritance pattern. So I'll write down diagenic inheritance pattern. Okay. Now what is it? Let me make you understand. For making you understand in a simpler manner, I will take a normal. Let me take a normal female for the first thing. Okay, normal female I'll take, and I will take an affected male. This is generally how you will be made understand. Books will also show you in the same manner. So I'll go according to that. So I'll take a normal male and I'll take an affected, normal female and an affected male. Okay. Now you just give me the genotype. So if the female is normal, she'll be like XX, nothing wrong. If the male is affected, he'll be XHO mutation over there. And Y is as such, Y has no dealing with the blood clotting factors. But this H means mutation has happened. She is normal. Now let us produce the gametes. Let us obtain the gametes. We're talking about the gametogenesis, okay? Let us get the gametes. To be simple, she'll be able to produce only one type of gamete. Agree or not? So we'll show the gamete will be containing X. Two types of gametes, usually, not usually, always. One containing X, another containing Y. But the problem over here is X has mutation. Okay. Now let us do the cross. Let us perform the fertilization. Let us see the fertilization consequence. Let us see the fertilization consequence. If this X fertilizes this X, what is the product? X, but one X is defective. Product will be a female. Will she be developing a disease? No, because the dominant gene is the homologous pair chromosome is that will do the job, that will produce the blood clotting factors. This female will not be affected, although she will be a carrier. Simple. If this X chromosome fertilizes this Y chromosome, the individual will be male. Okay, this will be a male as usual. No effect. So this will be a normal male. So in this case, there are 50% chances for the female to be affected and 50% chances for the male to be safe. This is how the probability will work if we take a normal female and an affected male. That means affected male cannot transfer this disease. You have to guess what has happened in this case. Affected male, that means father has passed on his trait of hemophilia that has gone to his daughter. That means the trait, the disorder has traveled from father to the daughter. Okay, so trait has come here, although she has not a disease. She is not disease, but she has become a carrier and she has obtained this trait from her father. Okay, so father to daughter. Okay, dear students, what I'll do is I will take up this carrier female, and we will take a case when she gets married with a normal male. Okay, you have to remember. So I'll be taking up this, carrier female. So I'll rub off this board. Remember the case. I'll be asking you question now once I'll finish it. Okay. Now, my dear students, I will take whom? I will take the carrier female. Let me take the carrier female. Okay. And let me take a normal man now. I will take a normal male. A male who is normal. Okay. Can you please give me the genotypes? X, H and X, correct? And male, X, Y, correct? Because he is normal. Let's take up the gametes. Gametes will be in this case 2 types. Because you have one X mutated. So one ova will be having defective X and another will be having the normal X. One sperm will be carrying X, another sperm will be carrying Y. So these are your gametes. Now let us perform the cross. Okay, in order to show you the cross, I could make a Punnett square. Now over here, since I have all the board clean, I can show you like this. Let me make a Punnett square that will look presentable. Okay, so like I'll make a Punnett square because that takes us less amount of space, and that becomes orderly also. So let us try and over here. Let us place the gametes. Please bear with me. Lines have become a bit tilted. So I'll place the male gametes this way and female gametes this way. Okay, female gamete I'll place on this side and male gamete I'll place on the vertical side. So male is X sperm, Y sperm. So these are the sperms. Okay, let me show the denotation. And over here I'll place the ovas. So one is the defective ova, another is the normal ova. Defective ova, normal sperm, X carrying, Y carrying, XX, because ovas are always X carrying. So now to do a cross. This with this sperm, this ova fertilize, this sperm, this ova fertilize. Now let's show this and this, the product falls over here. You might be knowing this is the Punnett square. This and this, X and X normal. This and this, X defective, Y. This and this, you have X, Y. Now look at the cases. Observe the condition. She is female, but she is a carrier because she has defect in one gene, one chromosome. Recessive form of mutation, because I already told you the gene itself is recessive. The mutation happens to be recessive. It's a recessive form of mutation. Remember those tidbits. So she will be definitely she'll be a carrier female. What about this? Definitely a normal female, carrier female, a normal female. Possibilities we are checking out on a Punnett square. Possibilities of the cross. Okay, Punnett is presentable. And then if we do this and this, what is this? Male. What will be the case? You just give me the answer. What is the case over here? Will this male be affected or not? Yes, definitely, ma'am, it will be affected because it has mutated X and there is no other alternative option available for checking out the expression of this defective gene. So he will be an affected male. How many organisms in this case affected? Just one, male only. And this, in this case, this will be a normal male. What is the probability distribution? Probability distribution is 25% chances for carrier female, 25% chances for normal female, 25% probability for getting an affected male, 25% probability for a normal male. This is the case. Or we can say that generally the male and the female normal cases will be 50%. That's another case to put it out. Now, what is the conclusion? The conclusion is that if you think about case 1, which was earlier, this is case 2. If you think from 1, when I take the affected father, you can check out your notebook if you have maintained with me. Affected father basically had this hemophilic mutation, basically a gene. He carried a mutated gene. He carried this gene. Okay, he transferred this defective gene to the daughter. Remember, his daughter, which is over here right now. He gave this to the daughter, and daughter became a carrier because she got one X. So she got one X from the father. Other X which she got was normal, which she got from her mother. Okay. Now, when this daughter was married with this individual who is normal, this daughter resulted in the formation of these many types of children, out of which we have seen that there is a possibility of one affected male. That means this daughter gave this defect to the son. Son was born affected, and because of whom? Not because of the father, it's because of the carrier mother. Son became XH and he got Y from father. So what is the conclusion over here? The conclusion is that the transfer takes place in which way? It takes place from the father to the daughter, pay attention. And then from daughter, this trait is sent to it's transferred, it's inherited by the son. That means from grandfather, it went on to the grandson. His daughter was just a carrier. This generation skipped the disease. That's why you also call it a skip. I got this point, okay? Skip generation inheritance. You also call it a diagenic inheritance, zigzag. Skip generation because this generation was skipped. You didn't saw the effect in this, and one generation was skipped. That the next generation, there is a probability that this child can be born. Okay, or it could happen that a female like this could be born, which could carry the disease. If she is married to some other normal individual, the case comes on. It prolongs. It remains in the family. It can only go because of a back mutation. Nothing else can change it. Only a back mutation can reverse, which has the least probability. Otherwise, the mutation remains there in the family forever. Generally, for this disorder, back mutation doesn't work. It preferably works with disorders like sickle cell anemia. Otherwise, they run in the families. Leave this apart. So, we have father affected, transferring this defect to the daughter becoming a carrier, and this carrier, if she gives her defective H to her son, the son will become affected. So you got to notice that the transference takes place in zigzag manner. That's why you call it crisscross inheritance. I will use another pen because we are going to conclude the lecture. So this is crisscross inheritance, zigzag inheritance, skip generation inheritance, the way you use it. And you will find many other diseases, generally X-linked diseases, follow this order because the logic is simple. Female has 2X, so only if 1X is defective, she is safe. Male has only 1X. If that is defective, he has. So who is more susceptible to X mutations, X-linked mutations? Male generally has no option. He will express the disease, and he generally will not live so longer, and he will die. He has not a normal lifespan. Okay, Females can also be hemophilic, but the chances are rare. When can female be affected with hemophilia? You can think, if this male is also affected with hemophilia, then this male, if his X is also affected, then we can think of this situation where this can also be affected and this female could be born. But what is this condition? I've told you, this is a lethal condition. This individual will not be born. Such females are not born. So generally in population, that's why you have only males affected with disease. Males transferring the disorder to a father transferring this disorder to the grandson, but through the daughter. So Queen Victoria, you call this as royal disease. Queen Victoria was what basically? A carrier. She had received this mutated X chromosome from her father. So her daughters also became the carriers. She transferred this mutation to the daughter. Okay, so daughter also became a carrier. Suppose she's a carrier female. If this X, defective X, as was the normal case, I'll remove this. So this was the daughter of what? Queen Victoria. She was married to, or we can say this was the daughter of Queen Victoria. She was married to a normal male, and then their descendants, the Queen Victoria's grandsons, great-grandsons, they developed this disease. And the situation you understood now, the carrier was Queen Victoria. That's why you call it a royal disease, because you find out this situation in her descendants. That means in royal family, the kings and the princes suffering from this particular disorder. This was all in this particular, video lecture. We completed hemophilia. Next is up to you people, whatever you want me to take up, the next genetic disorder. And one more thing, I'm planning for DNA fingerprinting and human genome project because they are very, very important questions. I'm basically busy with my personal stuff also. Still, I will try my level best to come up with the next video lecture as soon as possible. Take care of yourselves. Remember me in your prayers. That's what I need as a appreciation from you people. Remember me in your prayers. That's all for me. That's all what I need from you people. Allah bless you all. Fi amanillah.